Monoclonal antibodies have revolutionized the landscape of modern medicine, offering targeted therapies for a range of diseases that were once considered difficult or even impossible to treat. At their core, monoclonal antibodies are laboratory-produced molecules engineered to act as substitute antibodies that can restore, enhance, or mimic the immune system's attack on harmful cells. Their specificity and adaptability have made them invaluable tools in both diagnostics and therapeutics, bridging the gap between molecular biology and clinical application. The concept of monoclonal antibodies emerged from a fundamental understanding of how the immune system operates. Antibodies, or immunoglobulins, are proteins produced by B lymphocytes in response to antigens, foreign substances such as bacteria, viruses, or abnormal cells. Each antibody recognizes a unique epitope, or specific part of an antigen, through its variable region. In a natural immune response, a diverse population of antibodies is generated, each targeting different epitopes. However, this diversity, while advantageous for broad immunity, poses challenges for scientific study and therapeutic use, where uniformity and specificity are often desired. The breakthrough came in 1975, when George Kohler and Cesar Milstein developed a method to produce large quantities of identical, monoclonal antibodies. They accomplished this by fusing a single B cell, which produces a specific antibody, with a myeloma, cancer cell capable of rapid and indefinite division. The resulting hybridoma cell line could be cultured indefinitely in the laboratory, churning out vast amounts of a single type of antibody. This innovation not only earned them the Nobel Prize in Physiology or Medicine in 1984, but also paved the way for a new era in biomedical research and therapy. Monoclonal antibodies possess several advantages over polyclonal antibodies, which are derived from the serum of immunized animals and consist of a mixture of antibody molecules. The uniformity of monoclonal antibodies ensures consistent performance in diagnostic assays and therapeutic applications. Their high specificity allows for precise targeting of disease-associated molecules, minimizing off-target effects and reducing the risk of adverse reactions. Furthermore, advances in genetic engineering have enabled the production of humanized and fully human monoclonal antibodies, further decreasing the likelihood of immune rejection and improving their safety profile. In clinical practice, monoclonal antibodies have found widespread use across a variety of medical fields. One of their most prominent applications is in the treatment of cancer. Certain cancers express unique or overabundant proteins on their surfaces, making them susceptible to targeted antibody therapies. For instance, trastuzumab, Herceptin, binds to the HER2 receptor, which is overexpressed in some breast cancers, thereby inhibiting tumor growth and marking cancer cells for destruction by the immune system. Similarly, rituximab targets the CD20 antigen present on B-cell lymphomas, leading to the selective elimination of malignant cells. Beyond oncology, monoclonal antibodies have transformed the management of autoimmune diseases and chronic inflammatory conditions. Agents such as adalimumab and infliximab target tumor necrosis factor alpha, TNF-α, a cytokine involved in the inflammatory process underlying diseases like rheumatoid arthritis, Crohn's disease, and psoriasis. By neutralizing TNF-α, these antibodies reduce inflammation, alleviate symptoms, and slow disease progression. Other monoclonal antibodies have been developed to interfere with specific immune pathways, offering hope for patients with conditions that previously had limited treatment options. Monoclonal antibodies have also played a pivotal role in infectious disease management. During the COVID-19 pandemic, several monoclonal antibody therapies were rapidly developed and deployed to neutralize the SARS-CoV-2 virus, especially in high-risk patients. These antibodies bind to the viral spike protein, blocking its entry into human cells and reducing the severity of infection. Their rapid development and deployment underscored the versatility and potential of monoclonal antibody technology in responding to emerging public health threats. The utility of monoclonal antibodies extends beyond therapy into diagnostics and research. In laboratory settings, they serve as highly specific probes for detecting proteins, pathogens, or other molecules of interest. Monoclonal antibodies are integral to techniques such as enzyme-linked immunosorbent assay, ELISA, flow cytometry, and immunohistochemistry, enabling researchers and clinicians to identify disease markers, monitor disease progression, and guide treatment decisions with remarkable precision. Despite their many advantages, the production and use of monoclonal antibodies are not without challenges. Manufacturing these complex proteins requires sophisticated biotechnological infrastructure, including mammalian cell cultures, rigorous purification processes, and stringent quality control measures. The costs associated with production, coupled with the need for cold chain storage and administration via injection or infusion, can limit accessibility, particularly in resource-limited settings. Moreover, while monoclonal antibodies are generally well tolerated, some patients may experience infusion reactions, immunogenicity, or other side effects, necessitating careful monitoring and management. Ongoing research seeks to address these challenges and expand the potential of monoclonal antibody therapies. Advances in antibody engineering have led to the development of bispecific antibodies, which can simultaneously bind to two different antigens, enhancing their therapeutic efficacy. Antibody-drug conjugates combine the specificity of monoclonal antibodies with the potency of cytotoxic drugs, delivering targeted therapy directly to disease cells while sparing healthy tissues. Efforts are also underway to develop oral formulations, improve delivery methods, and reduce production costs, making these therapies more accessible to patients around the world. The future of monoclonal antibodies appears bright, with ongoing innovations poised to further broaden their impact. As our understanding of disease mechanisms deepens and technologies such as artificial intelligence and genomics advance, the design and selection of monoclonal antibodies are becoming increasingly sophisticated. Personalized medicine approaches, in which therapies are tailored to the unique molecular profile of each patient's disease, are likely to benefit immensely from the precision that monoclonal antibodies offer. In summary, monoclonal antibodies represent one of the most significant achievements in biomedical science, translating basic immunological principles into powerful tools for diagnosis, research, and therapy. Their specificity, adaptability, and proven clinical efficacy have made them indispensable in the fight against cancer, autoimmune disorders, infectious diseases, and beyond. While challenges remain in terms of cost, accessibility, and potential side effects, ongoing research and technological advancements promise to overcome these hurdles, ushering in an era where monoclonal antibodies continue to transform the practice of medicine and improve patient outcomes worldwide.